Hi, everybody. My name is Divyam Goyal, and this is my presentation for the fall 2022 iteration of UROP. Unfortunately, I wasn't aware that the requirement to present was before you graduate. I graduate this semester, and I was originally hoping to present at one of the research seminars later on, maybe in the spring when I would have more data to show, because as you'll see, my project over these last 2 or 3 months has largely been theoretical in nature, doing a lot of background work and a lot of tool making, I guess, which I'll explain later. So I don't have too much data to show as of now, but instead I'll use this opportunity to more talk about the background of my project, what we have done so far, some of the obstacles we've run into, and what we plan to do in the future. So my project heavily focuses on ST131 E. coli. So ST stands for sequence typing, and what it is a method of phylogenetically comparing different strains of bacteria, in this case E. coli. So E. coli are obviously very genetically diverse. I think an individual strain of E. coli can differ by as much as 30% in its total genetic material from another strain of E. coli, which is insane when you think that we are different from chimpanzees as humans by only a very small percentage, less than 10, I think. So what sequence typing is you basically look at variations in what are called 7 housekeeping genes, and bacteria that have the same pattern of variations in those 7 housekeeping genes fall under the same sequence type. So sequence type 131, ST131 E. coli, is of interest because it is a rapidly emerging, usually very antibiotic resistant strain of E. coli that is known to cause extraintestinal pathogenic disease. So ExPEC, which is extraintestinal pathogenic E. coli, is another categorization of E. coli, one of which the sequence types is ST131. ST131 is also known to harbor this CTXM15 set of genes, which are extended spectrum beta lactamases. So again, very antibiotic resistant. It's the most common form of ExPEC and is considered to be responsible for the most amount of antibiotic resistant E. coli infections in the United States. What's also interesting is that ST131 has been shown to outperform non-ST131 ExPEC in some virulence factors assays, specifically cell invasion and adhesion, which is really interesting because, again, ST131 commonly is very antibiotic resistant. It carries a lot of these AMR genes. And the idea is that something that is putting in so much effort and energy and resources into maintaining such a large set of extra genes might have to sacrifice virulence genes and other genes as sort of a fitness trade-off. But with ST131, we don't really see that. So that leads to some very interesting questions about this particular organism. Um, when it comes to, oops. So the lab I'm in, which is with Dr. Matthew Mulvey up in the Department of Pathology, they've done a lot of previous work on ST131. Specifically, they have been working with samples from one female elderly patient who's had recurrent UTIs or urinary tract infections since the 1970s. So that's over 50 years. And in 2012, they started doing some sequencing work, and what they found were these 5 strains here on the right side. Each of these strains is a urine isolate, hence the U, and the number that follows is for the year when it was isolated. So U12A is 2012, which was the first strain, and then 13, 2014, 2015, and so on. So what you can see is this phylogenetic tree shows that all 5 of these strains from this UTI patient are really genetically similar. All of these other limbs of the tree are ST131 isolates from around the world, and the ones highlighted in purple are ST131 isolates from the Salt Lake City area in Utah. So you can see that these particular 5 isolates from this patient are not geographically related to any of the other Utah ones, for example, but they are really related to each other. And the sequencing work that was done in this lab before I came along showed that at any point you would have these strains persisting regardless of the antibiotic treatment that this woman was receiving. So she's received all kinds of antibiotics over the last 50 years. She's received stuff that they did, sensitivity assays on to show that this antibiotic would work in vitro on this strain of ST131. But even though she would take it, she would have relief for 8 to 9 months, but the infection would come back. And so obviously that has been incredibly frustrating for this patient. And the sequencing work showed that after she got antibiotics, the population of ST131 that's colonized her gut and stuff did go down, but a very small amount of it persisted and eventually grew and expanded and reoccupied the niche that it's been maintaining for all these 50 years. So that's really interesting, right? With all the antibiotics she's been receiving, they're not able to put away this microbe for good. Another thing they found was that at any time when she's not on antibiotics, they found both antibiotic resistant and antibiotic sensitive strains of ST131, which were coexisting in the patient. So this is really interesting because it suggests that the bacteria are load managing the burden of these plasmids. It's been estimated that at any one time, about 1 to 5% of the ST131 isolates within this patient have these antibiotic resistance plasmids, which is a really small percentage. But what's hypothesized is that once the patient takes antibiotics at any time, and the bacterial population comes under antibiotic stress, the sensitive ones will die off, but the really small percentage that are carrying the load of those resistance plasmids, they kind of take over and replenish the health of the population. And then once the patient goes off antibiotics, the sensitive ones kind of take over because they don't have the burden of maintaining those plasmids, and the resistant ones carrying the plasmids kind of go back to their 1 to 5% rate. So that's a really interesting example of population dynamics with ST131, and maybe one of the reasons why it's able to survive, cause such devastating infections, and in the case of this one patient, persist for over 50 years. Let's see. So one place where I'm kind of going deeper is to look at the role of biofilms. So biofilms are a consortium of bacteria that are generally attached to a surface, and they're encased in what are called extracellular polymeric substances, which can include polysaccharides, protein, eDNA, or really all kinds of stuff. Bacteria use what's called quorum sensing, which is a way to sense if there's enough bacteria around before they start secreting these things. And then biofilms can also be polymicrobial. They can have multiple different species cooperating together. And what's interesting about biofilms is that they've been shown to offer tremendous protection from antibiotics. They're, the bacteria kind of encased in this coating, but also facilitate horizontal gene transfer, which is the exchange of genetic material between bacteria. And there isn't too much research on ST131's biofilm producing capabilities, but what is out there has suggested that they produce really great, robust biofilms, and potentially they might be better at producing biofilms than non-ST131 ExPEC or that extracellular pathogenic E. coli. So, biofilms are a natural state of growth for bacteria in our gut, and with all these other kind of attributes they carry, they might also be one of the reasons why ST131 bacteria are able to have such persistent infections, and for how they're able to transfer their genetic material around from that 1 to 5% group all the way to the rest of the population under antibiotic stress. So that's kind of where my project takes hold. One of the things we're going to be doing is we're going to be characterizing the U15A ST131 strain. So again, that's this one right at the bottom here, the 2015 isolate from, I believe, March of that year. And we're going to be looking at competition assays to see if it indeed does hold on to that plasmid or if it lets go, and if nutrient factors affect its whether it holds on to the plasmid or not. And I'll go into that in a little bit. Another thing that we're working on, which has actually been taking up most of our time this semester, is developing fluorescence tools. So our idea is, and I'll show you some photos later, hopefully if we're able to tag the U15A plasmid. So U15A carries many plasmids, but there's one specific one that has this massive, what's called an antibiotic resistance island, which is just a set of several resistance genes for multiple antibiotics. And what we're hoping to do is tag that specific island with the green fluorescent protein. So then we can actually see the movement of the plasmid between bacteria by seeing which bacteria start fluorescing green. And as you can see, there's several steps to that process. We tried once and we're currently working on it, and I'll show you some data from it in the next few slides. But this is definitely where most of the molecular biology has come in, and this is where we're facing most of our obstacles right now, because getting an extra tag into a plasmid has a lot of challenges associated with it. And then finally, going back to the biofilms, CFT 073 is another ExPEC. It's uropathogenic. It causes urinary tract infections. It's not ST131 though. So what we're going to do is hopefully, if we're able to get this green fluorescent protein into the plasmid, we want to grow biofilms with both of these strains together and see if we can track the movement of the plasmid. A really nice thing is that we have CFT that is already fluorescent red. So the idea is to see if we can see a green ring basically move into areas of red. And I'll show you some photos for that in a second. So the competition assays that I talked about, it's really interesting is, so here we use trimethoprim sensitive and trimethoprim resistant. So one of the many antibiotics that is whose resistance is encoded for on that U15A plasmid is trimethoprim. So the trimethoprim resistant are U15A, and then trimethoprim sensitive, this is actually another strain from the same patient that we were talking about who's had recurrent UTIs for over 50 years. It is also an ST131 strain. It is just one that doesn't carry that specific plasmid. So they're almost identical to each other. One has the plasmid and one doesn't. That's the only difference. And you can see here in LB, this is a ratio. So anytime the number is higher than 1, the sensitive has outcompeted the resistant. And anytime the number is lower than 1, the resistant has outcompeted the sensitive. So in LB, which is a very nutritious media, you can see that the sensitive one that doesn't have the plasmid is growing faster than the resistant. And then what's really interesting is M9 is a low nutrient and slightly stressful condition for the bacteria. And in that condition, at 72 hours, we've done this all the way up to 5 days. At 72 hours, so 3 days in, it looks like the U15A that is carrying the plasmid is slightly outcompeting the sensitive strain, which is really interesting. We've only done a few trials, so we're working on more trials before we can start running stats on this to get more data. But qualitatively, it does look like in kind of challenging conditions and stressful conditions that U15A really holds on to its plasmid and kind of refuses to let go. So that's one of the pieces of data we do have and has been really interesting to work off of. Again, so the molecular biology so far has been a challenge. We've had to make U15A electrocompetent. We're using a lambda red recombination system to get GFP in. So we had to first get a plasmid called PKM208 that has those lambda red genes into U15A. That we were able to do. That was pretty successful. And then we had to amplify GFP off of a donor plasmid. And this gel right here is that amplification. So you can see it looks like we have a little bit of contamination in the blank, which could be the reason why our first try was unsuccessful. However, because we had such a bright, fantastic looking band for our sample, we felt comfortable moving forward. But in the future, when we try again in these coming weeks, we might not go forward if our blank is contaminated again. But I just wanted to show you this gel to see kind of where we are in that process. So I also wanted to show you some of the microscopy we're doing to really see that fluorescence that I was talking about. So this is a wonderful echo microscope here, really high tech. And it has great feature where it kind of forms a grid across the plate and just takes little square photos and then it stitches them together. And so here you can see this is a lab strain that has some red fluorescent protein already in it. And here we're just testing out the microscope to see what it can do. And as you can see, wherever there is some colony growing on our streak plate, that same area is popping up red fluorescent back there on the photo on the screen. So, if we grow different strains on here, we can see different colors pop up. And that's really how we're doing the qualitative sort of visual part of this project, again, which is to see if that green plasmid eventually moves into areas of red. So here you can see we have the control strain. So this is the donor strain that already has the GFP that we're amplifying off of. This is what that looks like under the microscope. These lines, this is just some artifact. The program behind the microscope sometimes freaks out when there's a little too much light, and it can do that. And then this is our U15A after our first try. So as you can see, unfortunately, there's no fluorescence, which means the plasmid wasn't successfully tagged. So we'll try again. But this is that red CFT I was talking about. So you can see the CFT we have is ready to go, fluorescing a beautiful red. And hopefully what we want to see is little patches of green eventually move into the red to show that the plasmid has transferred from U15A to CFT. So that's what's to come. And then finally, we have this really cool photo of some biofilms. So this is from earlier in the year, actually several months before UROP. This is when I was playing around with the help of some grad students with biofilms, fluorescent biofilms on that microscope, just to learn more about microscopy and see what we can and can't do with it. And so this is two strains of that uropathogenic ExPEC, the CFT. One is green and one is red, and we kind of mix them together in the middle here and then just let them grow. And this biofilm, it's a really thick biofilm. You can see it's completely confluent. Compare this to these kind of streaky colonies here. This is like, very thick growth. But what you can see is they very interestingly branch out and into sort of like phylogenetic groups. I'm not really sure, but I've made this blue arrow to show where two of the colonies, two of the areas sort of intersect. And if we were to see any plasmid transfer, this is where it would be. So imagine if this green is that U15A and that red is the CFT, this little interface is where we would expect to see that plasmid transfer. So we can zoom in and kind of take more photos. But this was an example of that biofilm that we're going to be doing, hopefully, in the future once U15A is tagged, and really how beautiful it looks and how well the microscope works for this. So we're really hopeful. And we've got a lot more work to do, but hopefully this gives you an idea of where this project is going in the future, in the coming months, and what we hope to accomplish. With that, I wanted to thank the Office of Undergraduate Research for their funding, their support. I wanted to thank all of you for watching this. I wanted to also thank everyone at the Matthew Mulvey Lab, specifically O'Connor Matthews, a graduate student who's been working his butt off helping me with this. I really appreciate it. And I'm really excited to see this go forward. Thank you guys so much.